So we have reviewed the lateral ventricles and the ventricular system, and that gives us a good basis for where the fluid-filled areas are in the brain, and now we can start placing the anatomy around it. And this is your list of items to study for the cerebral hemispheres as well as a diencephalon. Knowing your midline structures can help you immensely when you start to place the anatomy from the cerebral hemispheres around that central anatomy. And we will look at these a little closer later on in the lecture, but the corpus callosum, the third ventricle with the intrathalamic adhesion, the septum pellucidum, the pituitary gland with the pituitary stalk or infundibulum, the pineal gland, the tectal plate, fourth ventricle. All of these things are midline structures, and when you start to look at those in MRI scans, on an axial scan or a coronal scan, even sagittal scans, and knowing what is midline and then what is to either side of that midline, going out further and further in the brain as you go laterally and anterior and posteriorly, you're able to better then place the anatomy around those midline structures. Along with that, then you are also able to see what normal brain tissue looks like, and then when things are slightly askew or when things are being pushed to one side or another, then you're able to see that there's might be an underlying cause for that, or see what the abnormality is. So know these midline structures. These are two great images. This one on the right is from Netter's 6th edition, plate 107. And then this one on the left is from eAnatomy, and this is in the 3D brain, which is only in T1 weighted. But we're going to take a look on this next slide of both the T1 and the T2 weighted slices from eAnatomy. I find the T2 weighted images really helpful in taking a look at the gray-white matter differentiation, especially deep within the brain. When you take a look at these two slices side by side, and these are both from eAnatomy, you can take a look at the bright white CSF of the lateral ventricles. So this is the anterior horn of the lateral ventricles coming into the third ventricle here, and then the occipital horns of the lateral ventricles. And you can see how bright white this is. It's a little slightly different slice here on the right with the T1 weighted, but you can still see the dark of the CSF. You can see where the anterior horns would be coming into the third ventricle here, and then the occipital horns of the lateral ventricles. And with the gray and white matter, the best thing to do is to take a look at the cortex. The cortex is going to be gray matter, and it is on a T2 weighted, it is a slightly lighter gray shade than the white matter. So you start with the cortex and the gray matter, and you can tell what the deep set gray matter is going to look like by looking at the cortex. Same with the T1 weighted, you can see how the cortex is slightly darker gray, and when you look for that and then you look at the deep set gray matter, you can see how that is a slightly darker gray. And going back to the T2 weighted, so you have the slightly lighter gray of the cortex, and then you have the deep set white matter, and this is, the white matter tracks, and this is a darker gray. And so you can tell that this is white matter here, white matter along here, and along the outside of this gray matter. Those are all white matter tracks. And the gray is where the nuclei sit, and they're where the initial pulses of information are sent from. And then the white matter tracks are like the highways where the information travels onto. So information is being passed from gray matter to gray matter area, and they go through the white matter to get there. And then the deep set gray matter, this is the head of the caudate nucleus, which we'll study a little bit more in a bit. And then this is the putamen right here. And this is all part of the basal ganglia. In all of these lectures, I try to bring up the pictures from eAnatomy and other CT and MRI scans, as well as the illustrations and pictures from Netter's, both Netter's and eAnatomy, that help me to visualize the anatomy the best. There are so many illustrations and images in all of these places for you to look at, as well as on Google Images or Bing, whatever your search preference is. And I encourage you to research all of them and spend some time browsing, because what is obvious to me or what makes a lot of sense to me when I look at the images may not work for you. So take a look at all of the images and see which ones help you the most when studying for the exams and also just studying in general to get to know the anatomy. I'm taking a look at this image from e-anatomy. We're going to take a look at the hemispheres, and we're going to start with the frontal lobe. And the frontal lobe here in sort of a Seahawks green, if you will. Nice and bright, and it's the most anterior and the most superior lobe as well. So this is the frontal lobe, and there is a central sulcus here that divides the frontal lobe from the parietal lobe, and the parietal lobe is posterior to the frontal lobe. And then from there you go to the occipital lobe, and the occipital lobe is the most posterior lobe. And then the temporal lobe is the most inferior lobe. It is also the most lateral lobe. There's a left and a right, of course, and it sits the most lateral in the brain. And then there is a lateral sulcus here, also a Sylvian fissure that separates the temporal lobe from the parietal and the frontal lobe. So it separates all of those lobes here. And just deep within this temporal lobe is in the insula or insular lobe, and that sits just medial to the temporal lobe, and we're going to see that better on an axial slice. So this is a similar view from Netter's 6th edition plate 106, and you can see the various hemispheres of the brain. And then also in this image here on the right, Netter's has pulled down the temporal lobe and pulled up the frontal and parietal lobes, and so that you can see that insular lobe or insula deep to the temporal lobe. So that's a nice image and a nice way to see that. And again, on the axial slice, this will make a little more sense to you as to where that is positioned. So while we won't be going this deep into our studies for this class, these are nice images from Netter's to show you where the motor sensory locations are within the brain along the cortex. The association pathways. This is a motor sensory area here, and then sensory motor here. You've got the visual back in the occipital lobe, the auditory here along the temporal lobe, and then a little bit deeper within that, the olfactory or sense of smell. So this is just kind of fun to look at, and maybe an image that you may want to draw on later in your studies if you go deeper into anatomy and physiology. On the right here we have the image of the brain from e-anatomy, and this shows you how the ventricles sit within the hemispheres of the brain. So you have the frontal horns of the lateral ventricles within the frontal lobe, and then the body of the ventricles within the parietal lobe, the occipital horn within the occipital lobe, and then the temporal horn within the temporal lobe. So you can see how that pattern and that shape follow the shape of the brain. And this shape will start to become very familiar to you as the caudate nucleus also follows this shape, as does part of the fornix also follows this shape that we'll see later. So on an axial slice, this is an axial T2, through the temporal lobes, and we are able to see the frontal lobes here, and the cerebral falx creates that fold that separates the frontal lobes anteriorly, and the cerebral falx runs from the superior sagittal sinus to the inferior sagittal sinus. Again, the two hemispheres of the brain should be symmetrical, and if they're not, you can take a look and see if there's something going on, what the abnormality might be. But that cerebral falx separates the frontal lobes, and then we have the temporal lobes here that are laterally. The parietal lobe would sit about here, but that again is superior to this slice. And then we have the occipital lobes as the most posterior lobe of the brain. And then separating that frontal lobe and parietal lobe from the temporal lobe is a Sylvian fissure, and you can see that all of these sulci are bathed in CSF. So that's easy to see here because the CSF is bright white. So you have the left Sylvian fissure here and the right Sylvian fissure here and the temporal lobes, and then just deep to the temporal lobe is the insula in the insular region, and you can see just deep to that is the deep set gray matter, and that's just internal to the insula on each side. So this would be the left deep gray set matter and the right deep set gray matter, and we will go over the details of that in just a moment. Now visualizing that in a T2 weighted coronal MRI scan, we have the superior area here, inferior, right, and left. And these are the parietal lobes here. And you see that central sulcus, which is the falx that is separating the right and left lobes of the brain. And this right here will be the corpus callosum. This is a white matter tract. You've got the anterior horns of the lateral ventricles, the right and the left. And then we have the temporal lobes here, the right temporal lobe, the left temporal lobe, the sylvian fissure on the right and on the left. And then just deep to that is the insula, left insula, right insula. So this is a good visualization of that on a T2 weighted coronal image. Remember, the cortex has the gray matter, and then deep to that is the white matter, and then we have the deep set gray matter, which is a little hard to visualize in this particular image, maybe due to the resolution of this particular scan or the slice thickness.